The next phase in meiosis 1 after prophase 1, that is the metaphase 1. So what are the events that are taking place in metaphase? This is a very simple phase in meiosis 1. We have seen in the prophase 1, the spindle formation has taken place and the chromosomes, they are present inside the nucleus. But all these chromosomes, they are brought on the equatorial plane. This chromosome set is above on this side of the pole. This chromosome set is near to the equator. So it is perfectly brought to the equatorial line during the metaphase. What does it mean? The homologous chromosome pairs, they are aligned. They are aligned at the equatorial position. Equatorial position in case of metaphase, and this is the only important event in case of the metaphase. How it is brought? With the help of, again similar to the mitosis, with the help of these spindle fibers, they will contract and this pair it will come down. This pair, again this spindle fiber, it will contract and this is brought at the center. So how it appears, that we will see here. With the help of this contraction, the homologous pair of chromosome, it is brought at the equatorial level. At the equatorial level. Here how it appears like this. This is the chromosome, this is the kinetochore, and fiber is attached at this point, and here one more pair it will be there. So again, this is the exchange area, that's why I'm taking it like this. This is the thing and this is the thing. So this. So this spindle fiber, it is contracting so that the chromosome, it will come at the equatorial point. Again this has contracted, the homologous chromosome pair, it has come to the equatorial level. That is seen in case of the metaphase 1. The next phase after the prophase 1 is the metaphase 1. In this metaphase 1, the homologous chromosome pair, they are brought to the equatorial position. That is alignment of the homologous chromosome pair at the equatorial position. That is the important event that is taking place in case of the metaphase 1. If you just have a look, it has been aligned at the equatorial position by contraction of the spindle fibers. Contraction of the spindle fibers. Next to the metaphase 1, next phase is the anaphase 1. What is that? That is the anaphase 1. What are the important events that are going to take place in the anaphase 1? That we will see here. In anaphase 1, the chromosomes, they are taken. These chromosomes, they are pulled towards the opposite poles. They will be pulled with the help of fibers so that one chromosome from this and one chromosome from this point, it will be moving towards one pole, and on another pole these two chromosomes, they will be moving towards the other. That is happening in the anaphase. But when we talk about the anaphase 1, it is something different from anaphase in case of mitosis. What is that difference? If you just have a look in the mitosis anaphase, you are seeing this is a centromere, this is a kinetochore, and these are the spindle fibers. So when it is pulled on the opposite direction, there is a splitting of centromere it has taken place. I'm talking about the anaphase in mitosis. What is different in anaphase 1 of prophase 1 in meiosis? Here, the spindle fibers that are attached to the kinetochore are the complete chromosome, one chromosome among the homologous chromosome. And another spindle fiber is attached to the another chromosome. And when they are pulled on the opposite direction, when they are pulled in the opposite direction, this chromosome it will move towards one pole and this chromosome will move towards another. But there is no splitting of centromere. It is there. No splitting of centromere because entire chromosome it is taken there. And in this you will find there is a sister chromatids that are present. Entire chromatid it is taken to one pole. Entire chromosome it is taken to another. So this is going to happen in the anaphase 1. If we just have a look, this will be the picture how it will appear. And when this particular thing it is moving, when this particular chromosomes they are moving, and when this movement of the chromosome it is taking place towards the opposite poles, in between these two there is no any fiber that is present. If you recollect, in the anaphase of mitosis, when splitting of centromere it takes place, if the splitting of centromere it is taking place, this is centromere, this is centromere, this is how it is taking place. In between these two there was one fiber that is seen. What was that fiber? This fiber it was known as interzonal fiber. That was the case in mitosis when the centromere splits, but this is not the case in case of anaphase 1 of meiosis. No fiber it is seen in between. In between there is no interzonal fiber it is seen because no splitting of centromere it has taken place. I hope that you might have understand. We have seen this anaphase 1, and this is the place where the chromosome they are divided into half. From 2n is equal to 4c chromosomes they are present. 2c it is carried, if you just have a look towards this 2c it is carried towards this pole, and 2c they are carried towards this pole. So here if you just have a look, one n chromosome and another n chromosome it is splitted in the anaphase 1. The last phase of meiosis 1 after anaphase 1 is the telophase 1. Is the telophase 1. So what exactly it is happening in the telophase? If you just have a look, these chromosomes they will reach towards the pole. That is one thing that is going to happen. So I will describe here what is going to happen. So what is going to happen in telophase 1, the last phase of meiosis 1? What is going to happen? The chromosomes they have reached at this point, and the chromosome they are getting decondensed. You will find somewhat thin chromosome or chromatin fiber like structures here at the opposite poles. Centriole it is present, centriole it is present, spindle fibers they get off, disappeared, spindle fibers they get disappear, aster rays they get disappeared, chromatin, these chromosomes they become decondensed to form chromatin fibers, and they will reach at the opposite poles. That is going to happen in the telophase 1. Apart from this, the reformation of nuclear membrane it appears, reformation of nuclear membrane, that is seen in this, and reformation of other membranous organelles also will take place. Similarly, formation of notch, it is seen at this point, formation of notch, that is seen at this point. So these are the things which are taking place in telophase 1. What are those? The one chromosome, two number of chromosome, they are reached at this pole and they have got decondensed. They are turning towards the chromatin fibers. Apart from that, nuclear membrane, it reappears. Nuclear membranous organelles, other membranous organelles also they reappear or reform. Already the other membranes, they are also forming. Already the centrosome it is present. Already the centrosome it is present, and there is a formation of furrow that is taking place here. So in telophase 1, so this is how the karyokinesis it has taken place. After the karyokinesis, it comes cytokinesis 1. What happens in the cytokinesis? Already the furrow formation it has taken place. This furrow it will deepen. It will deepen and two daughter cells. Two daughter cells they are formed separately. They are formed two daughter cells. They have formed separately. These are the two daughter cells which are formed. This is one and this is another one. So two daughter cells they are formed in the cytokinesis. It's a very important point. And these two daughter cells they are of one end and another end. Here two number of chromosomes 2C and 2C here you will find. Generally when we talk about the cytokinesis, at the end of meiosis 1, after cytokinesis, from a single meiocyte which was 2N earlier, it has given two daughter cells with single N and single N. That is the result of meiosis 1.